Welcome back, dear students. Today our topic is complex tissues, or you can say complex permanent tissues, because the individual cells of these tissues have lost the power of division. So let's move on to how you can first of all define complex permanent tissues. A group of more than one type of cells having common origin and functioning together as a single unit comprises a complex tissue. And in vascular plants, there are two important types of complex tissues. One is xylem, which is involved in the conduction of water and minerals from roots to shoots, and another one is phloem, which is associated in the translocation of food materials which are synthesized in the leaves and transported to different parts of the plants like roots, fruits, etc. Okay, here you can see in the image this region of the stem, okay, which appears in the form of rings. It is the secondary xylem and it is involved in the translocation of water. And outside this secondary xylem we have the secondary phloem, which is attached to the bark, okay, and this secondary phloem is involved in the translocation of carbon food, okay, or you can say organic food from the leaves to the roots and other parts of the plants. So let's move on first of all with xylem. The term xylem was introduced by Nageli in 1858 and it is derived from the Greek word xylos, which means wood. And xylem is the principal conductive tissue of vascular plants. I have already told you xylem is involved in the translocation, in the transport of water and minerals from roots to leaves. Now the components of xylem, and the first important component of xylem that we are going to discuss today is tracheids. Tracheids are elongated tube-like dead cells with tapering ends, and these cells, they are narrow, okay, they are narrow and they possess a lumen inside. But you have to remember that the lumen of tracheids, it is narrow as compared to the lumen of vessels, okay, and in cross section they appear to us as angular, polygonal, or in some cases they appear rounded. Here you can see these are tracheids which are long, tubular, and possess the tapering ends. If you will cut the transverse section of a stem, they will appear to us like this, okay, they will appear to us round, angular, or they will appear to us polygonal, or in some cases they will simply appear as rounded. Here you can see this is the image of the tracheids. These tracheids are long, they have tapering ends, and the ends of different tracheids, they are joined to each other with the help of bordered bits that we will discuss in the next slide, okay. First of all, let's discuss some other characters of the tracheids. Their walls are hard and lignified, but much of their inner space, it is not lignified, and because of this they possess a wide empty lumen, okay. But one thing that you will remember, the lumen of tracheids is narrow as compared to the lumen of vessels, okay. They possess hard and lignified walls, but their walls are not much thick, and because of this they possess a wide empty lumen. They are long and usually they measure 1 millimeter in length but can reach up to 12 centimeters in some plants. But you have to remember that the tracheids are not as long as are the fibers of xylem, that we will discuss in the next parts, okay. The tracheids of primary xylem develop from procambium, which is the embryonic meristem, but those of secondary xylem they develop from the vascular cambium, okay. जो secondary xylem में tracheids होते हैं, they develop from the vascular cambium. In the beginning these cells possess living protoplasm, but with time, due to lignification, they become dead at maturity. They lose their protoplasm, they lose their nucleus, okay, and become dead at maturity. Now, the pattern of wall thickness in tracheids. There are different types of tracheids based on how the secondary thicknesses are deposited, okay, depending upon the manner in which the secondary thicknesses are deposited. The first one is annular. Annular means ring-like. You can here see the secondary thicknesses are deposited in the form of rings. Second one is helical or spiral. They are deposited in the form of spirals. Third one is scleriform or ladder-like. The secondary thicknesses are deposited in the form of ladders. Okay, here you can see they are deposited. These white patches represent the secondary thickness. Okay. Another one is reticulate or network-like. The secondary thicknesses are deposited in the form of a network. Here you can see in the image, some patches are left undeposited and others are having the depositions of the secondary material. And the final one is pitted. In this type, small pits are left in the secondary wall while the rest of the secondary walls are having the depositions of lignin. These small pits are without the secondary depositions. Okay. And the pits are of two types. One is known as simple pit and another is known as bordered pit. In simple pits, here you can see in the image, you do not have any overarching of the secondary walls, while as in case of the bordered pits, there is overarching of the secondary walls on the pit cavity or pit aperture, you can say. Okay, the secondary walls, they overarch, okay, they overarch the pit aperture. This is known as bordered pit. Now the functions of tracheids. Tracheids are the principal components along with the vessels, okay, and they help in the conduction of water and minerals from roots to shoots. You know that tracheids are placed one above the other and their end walls are perforated by bordered pits, and because of these bordered pits, they allow the transport of materials, the transport of water and minerals from one tracheid to another. Let us make it more clear. Say this is one tracheid and this is another tracheid, okay, and they possess the tapering ends. Here at this point you have a bordered pit. At this point, let's make it more clear, it is not that much clear. Here you have one tracheid, we have another tracheid here, okay. At this point, at this tapering end, this possesses a bordered pit and this also possesses a bordered pit, okay, and these bordered pits, they form a bordered pit pair, and because of this bordered pit pair, there is translocation, there is flow of materials, water and minerals from this tracheid to this tracheid, okay. In addition to conduction of water and minerals, they also provide mechanical support to the plant body. They make it hard, okay, because of the firm secondary walls which contain lignin. Now some important remarks about the tracheids. The tracheids are present in all vascular plants. They are present in pteridophytes, gymnosperms and angiosperms. When we will discuss the vessels, you will come to know that vessels are only present in angiosperms, okay. This is the important defining feature of tracheids, okay. This is the characteristic feature of tracheids. You can see that tracheids are present in all vascular plants, pteridophytes, gymnosperms and angiosperms. These cells are elongated and they possess tapering ends, okay. This is the second important feature. Their end walls remain intact and possess bordered pits. जो इनके end walls होते हैं, I have already told you, they are intact and they possess bordered pits, and because of those bordered pits, they form the continuity and help in the transport of material from one cell to the another cell, okay. And they possess a narrow lumen as compared to the vessels, which we are going to discuss in the next part, okay. This is all about tracheids. I hope you all understand this.